Hello, and welcome to our lecture on Chapter 3. Welcome to our lecture on Chapter 6 on experimental research. In this lecture, I'm going to talk specifically about internal and external validity, starting with internal validity. Internal validity is the ability to have the confidence that our independent variable has actually caused the change that we are observing within our dependent variable. For example, let's say that you're studying individuals and their ability to help one another. You believe that sadness specifically leads to helping because it makes individuals feel better. Now, if you had conducted your study on September 12, 2001, how do you think this would influence your results? That is, now are you sure that the results that you have relate to how much people help one another? Is that related to sadness just due to September 11th events, due to your independent variable, or due to some confluence of the two events? Whenever you conduct a study, you have to be careful about confounding variables. Those are variables that can come in and obscure your independent variable and dependent variable relationship. When you have a confounding variable, you have very low internal validity. You need to work on protecting your internal validity. There's two specific procedures that help any researcher increase their internal validity. That is, to produce what we call control procedures. Control procedures are those that essentially help prevent our confounding variables from becoming part of our study. The second approach is to use what we call standard procedures. And standard procedures are experimental designs that are designed specifically to ensure our internal validity. With these procedures, you have to develop a plan for selecting your participants, for assigning your participants conditions, for controlling extraneous variables, and for gathering your data. At this point, you may wonder, well, this is just another concept related to research, right? No, it's not. Internal validity is actually the most important aspect of creating a research project. The other type of validity that I mentioned I talk about in this lecture is external validity. And external validity is the ability for your results to apply to other situations. In other words, how well do your study results generalize, that is, apply from your experiment to a different situation or population to the real world? When we think about how we generalize results, we typically do so in one of three ways. We either apply the results to a new population, a new environment, or a new period in time. So population generalization is the ability to apply results from your experiment to a group of participants that is different than those used in the original experiment. For example, in your third study, you will be creating an experiment where you probably will be utilizing Mount St. Mary students. Now, if you want to be able to engage in population generalization, you would be generalizing the results of your study done on Mount St. Mary students to perhaps women in general, perhaps adults in general. In other words, you're trying to go from your specific participant group to some larger, more encompassing group. Another method of generalizing includes environmental generalization. That is, applying the results from your experiment to a situation or environment that differs from the original experiment. For example, let's say in your third study that you are producing results related to memory, and you're doing so within a classroom. That is possible for you to do in your third study. However, if you would like to generalize these results to the way people remember stuff in the supermarket, or the way people remember items while having conversation with friends, those are environments that are different from the classroom, and you need to think about how well your results actually generalize to this new location. The third type of generalization that researchers typically engage in is temporal generalization. That is, applying the results from your experiment to a time period that is different from when the original experiment was conducted. Much of the research on obedience was done in a time prior to institutional review boards and other ethical standards that we have in place within psychology. However, we still believe that people are as obedient as they were in the 1940s, 50s, and 60s when those studies were conducted. The nature of the studies revealed something that is true about humanity across time. Now, if you're doing a study related to gender roles is something that has evolved over time and has specific cultural implications. If you did a study on gender roles in the 1920s, that would be very different in terms of the results than it would be to conduct these studies within the 1990s or within the early 2000s. Gender roles have changed over time. So the ability for a study's results on gender roles in the 1920s to apply to those in, let's say, 2010, we would probably lack some temporal generalization. That's not necessarily a bad thing. It just means that cultures have evolved over time. You need to think about whether you desire the ability to engage in any of these three types of generalization. Assuming you'd like to generalize your results in one of these three ways, how do you ensure you can do that? Well, there's very specific threats to external validity, and these are based on methods. So we have interaction of testing and treatment, the interaction of selection and treatment, reactive arrangements, demand characteristics, and multiple treatment interference. The interaction of testing and treatment occurs when we have some sort of pre-test in a research situation, and that sensitizes participants to the treatment that will come afterwards. Let's say that you're doing a research study on how effective a textbook is. And prior to showing people the textbook, you give them reviews that are already created by other professors. And if those reviews are mainly positive, what happens is they may sensitize participants to believe that the book is actually better than it is. Whereas if those statements are generally negative, participants may view the book more negatively as well. How do you deal with the pretest that sensitizes participants? Well, one of the things that you can do is when you design the study, you can do what we call a randomized post-test control group design. And within a randomized post-test control group design, what we do is we randomly assign participants to a condition, either our treatment condition or our control condition. And then when we measure them only at the end of the study. So rather than giving people the reviews ahead of time of the textbook, all we would do is we would present the textbook to our participants, and we would engage in, let's say, maybe a full color version versus a black and white version. And then we measure people's reactions at the end of the test. By eliminating the pretest altogether, you remove the ability to have this pretest sensitizing effect. Another way to control for the effects of a pretest would be to engage in a randomized pretest post-test control group design. And in this design, what happens is you randomize people to groups, either our treatment group or our control group. And we give both groups the same pre-test. So by giving them the same pre-test, let's say we give them all positive evaluations, and then we give them the book in either black or white, in either color or black and white, and then measure the reactions at the end, we've eliminated the effect of different pre-tests. One of the best designs to actually deal with a sensitizing pretest is what we call the Solomon 4-group design. And within the Solomon 4-group design, participants are randomly assigned to one of 4 groups. In the first 2 conditions, they receive a pretest, but in the third and fourth conditions, they do not. Then, after the pretest is administered to those first 2 groups, and after time has elapsed in our third and fourth groups, they receive either the treatments or the control, that is, the colored books or the black and white books, and then they're measured on their dependent variable here in a post-test. What's nice about the Solomon 4-group design is that if you find that these scores are significantly different than these scores, you know that it's the pretest here that's causing the difference. So essentially, the Solomon 4-group design is the perfect answer to finding out whether your pretest has any sensitizing effects, and if it does, how those effects actually matter to your dependent variable observations. So that's how we deal with interactions of testing and treatment. Let's move on to interactions of selection and treatment. Interactions of selection and treatment occur when a treatment effect is found only for a specific sample of participants. And this is essentially what happens when one group, and only one group that you've selected for your experiment, is affected by our treatment. Interactions of selection and treatment are more common when you have difficulty finding participants. So for example, if you're studying something that's fairly rare, let's talk about maybe synesthesia, where people have extrasensory connections in their brain. So when they hear something, they also taste something at the same time. If you're working with a group that's already fairly limited, you may have more problems trying to assess treatment effects because you've got such a limited pool of participants already. Now that we've talked about what happens when we have a small population, let's move on to reactive arrangements. Reactive arrangements are caused when an experimental situation alters our behavior, irregardless of our independent variable that's involved. Essentially, reactive arrangements is just the exposure to the experiment itself. Just being part of an experiment has caused differences in our dependent variable. And now we don't know if we can actually generalize those results to the outside world, because we don't know, will these behaviors that we saw within our experiment, do they actually occur in the real world? Because we don't know if it's due to our artificial conditions, the lab in which we conduct our study, because those situations may not actually exist within the real world. So reactive arrangements occur when we have this lack of environmental generalization, the inability to go from our lab to the real world. For example, if you were studying the behavior of couples within a laboratory, how couples actually behave when in love, when they're being watched by a researcher, is probably different than how couples actually act when, let's say, they're in their own home. So that would be an example where we may have a reactive arrangement due to being in the laboratory study, and it's not due to the independent variable that's being exerted here. It's just due to the fact that they're in a laboratory setting having an experiment conducted. Let's move on to demand characteristics. Demand characteristics are features from our experiment that inadvertently lead participants to respond in a particular manner. One very common demand characteristic that we face within research is the fact that we deal with volunteers. So participants tend to volunteer their time to be part of research. Now, volunteers inherently are a special group of people. They are more willing to please, they're interested in research, they tend to be more highly educated. They tend to also try and figure out what's going on in the study in order to kind of help along the study. And because they are just nice people, this is inadvertently leading our results down a certain path. And now it becomes hard to generalize results to all adults, not just adults who are willing to be volunteers. So why this is a threat to external validity is because it makes generalizations more difficult. We don't know if we can apply results to the real world. Let's move to our last threat of external validity, which is multiple treatment interference. When you have a study that people are experiencing multiple treatments, that is a repeated measures design, you have this problem where our findings may only occur when the participants are experiencing those multiple treatments. Because a laboratory setting is set up to have very rigid procedural situations, we find that participants may have results due to experiencing these multiple treatments in a certain way within our laboratory. When we go to generalize these results to the real world, what we find is that because the real world does not mimic those carefully controlled procedures, the results end up being different. Therefore, we lack the ability to generalize the results from the laboratory setting with these carefully controlled procedures with multiple treatments to the real world. So I've talked through a few threats to external validity. Now keep in mind, we have to have internal validity, but we do not necessarily have to have external validity, although it can be a desirable trait for your study. With that in mind, I'd like you to consider how you act. Think about the information you receive from other research. Now, keep in mind that most of the research that you've seen is done on typically, originally, animal studies before moving on to human studies. And even if the studies that are leading you to make decisions about your life and have influenced maybe your education or your medical treatments, if they were done on humans, they were probably done on college students, which may or may not be representative of all adults. Also, we know that most of the research that is conducted is typically conducted on Caucasian Americans. That is, people from what we see as the typically majority culture within America. Do results related to white Caucasians apply to all other cultural backgrounds and all other ethnic backgrounds? Probably not, right? And the last threat that I'd like you to think about is that most of the research that you utilize is also conducted on men. Given that we're at a predominantly female institution, it may surprise you to learn that whenever we use rats, humans, typically in medical research, including that related to psychiatric care, it's typically male rats or typically male participants, because there is a mistaken idea within research that the female cycle would influence results related to drug research. Of course, the big caveat here is that when we do drug research, it's tested on traditionally male rats and then traditionally male participants. But who ends up taking the drug? Well, of course, men and women, right? So there's a larger push now to incorporate more variety within our research participants in order to have better external validity to the real world.